Hello everyone, I hope you are well. Welcome back to Understanding Cancer Season 2, Lecture 5, titled Signal Transduction and Cellular Response. So last lesson, hopefully you should have understood that the receptor activation is the first step of the cell-to-cell communication. The ligand is the first messenger that could be a protein or a steroid and can complementary bind with the receptor like a lock and key. Some receptors are found on the cell surface and other receptors are found inside the cells, i.e. in the nucleus and cytoplasm. Some ligands are hydrophilic, for example proteins, so they are water-loving, and cannot diffuse through the plasma membrane due to their size, so they require receptors on the cell surface. Other ligands, on the other hand, are hydrophobic, so they do not like water, for example steroid hormones, and therefore they are able to diffuse through the plasma membrane and interact with intracellular receptors. So intracellular means inside the cell. So the rate of the binding between a ligand and receptor equals the rate of releasing the ligand from the receptor. So what will we learn today? We will learn what is signal transduction, the importance of protein kinases, what are secondary messengers, examples of secondary messengers, how do cells respond, what are transcription factors, and types of domains in transcription factors. So a gentle reminder that this lecture will take no more than 1 hour in total. However, you are able to divide the lecture across the week and then you'll be able to challenge yourself with a quiz, so please work at your own pace. Just as a recap, and to support your learning, there are key facts with diagrams by HN Designs, so these designs you will be able to see them anywhere elsewhere, and the aim is to present it in a simplified way. The glossary is to help understand what keywords mean. Semi-doodle revision posters by HN Designs, and quizzes to be able to test your knowledge and reflect, and there is also a reference list for further reading. A special thanks to my parents, my family, my friends and colleagues for their support, and the respected teachers and health professions who taught me and instilled the passion of cancer. So let's begin with what is signal transduction. So signal transduction is the second step in cell communication. So last lesson we discovered receptor activation, and the aim of today is to look at signal transduction and cellular response, but we will begin with signal transduction. So what is signal transduction? So signal transduction occurs when you have a ligand or the signal, and it's been activated via the receptor. This signal then passes through the plasma membrane. So the plasma membrane is the barrier that controls what enters and leaves the cell, and therefore the signal is able to pass through to the cytoplasm. So when it enters into the cytoplasm, this is now known as the signal transduction pathway or the cascade. We will now look at the importance of protein kinases. So what are protein kinases? Protein kinases are enzymes. Okay, so enzymes are a type of protein, and protein kinases, they help to transmit the signal inside the cell, and this is via a process called phosphorylation. So phosphorylation is when you add a phosphate group, and dephosphorylation is the opposite, and when you want to dephosphorylate protein kinases, this can be achieved by protein kinase inhibitors or another enzyme called phosphatase. So when we discuss about phosphorylation, sometimes this actually activates the enzymes, sometimes this actually deactivates the enzymes. However, in the case of activating the enzymes, it can affect the activity of the enzyme, or it can create a binding site that interacts with downstream targets. So downstream means the later on within the signaling pathway. So let's look at an example over here. So in the previous lesson, we discussed how ATP and GTP are formed. So these are types of nucleotides. So when you have a phosphate being attached to an oxygen on the amino acid on a particular protein, how that occurs is when you remove a hydroxyl group, and a hydroxyl group is a group that contains O and H, so oxygen and hydrogen. So the OH needs to be removed in order to place this phosphate group. And examples of the amino acids that are phosphorylated within the particular protein is serine, threonine, and tyrosine. So there are many amino acids. So these are the three main examples of when we say phosphorylation of proteins. These are the amino acids within the protein that gets phosphorylated. So in this diagram you can see it's the phosphate group has been highlighted and where it's basically been attached to the actual amino acid. So what are secondary messengers? So let's have a recap of what are first messengers. So the ligand or signaling molecule is considered as a first messenger. So this could be a peptide hormone, for example, that can bind to a receptor on a cell surface membrane, or it can be a steroid hormone that are able to diffuse through the membrane. What are secondary messengers? So secondary messengers, they are small in size, non-protein, and it's basically used to help transmit or transduce the signal within inside the cell, so in the cytoplasm where the chemical reactions take place. And this, how it helps to transmit the signal is by changing the behavior of particular proteins. So let's look at examples of secondary messengers. Examples of secondary messengers are as follows: calcium ions, cyclic AMP, diacylglycerol, and inositol triphosphate. We will start off with calcium ions. So calcium ions is a common secondary messenger, and it has many important roles within cellular processes, and which we will discuss later on. So in this image over here, you can see that there are channels or pumps in how the calcium ions are able to enter, and there is some between the plasma membrane, and there is some in the mitochondria where respiration or the production of the energy takes place, and there is also within a structure called endoplasmic reticulum, which we will discuss later. So calcium ions are actually stored in structures called vesicles, and they are particularly involved in signaling pathways of water-soluble molecules. Now the aim of the cells is to maintain a low concentration or low levels of calcium ions within the cytoplasm, and in order for that to take place it needs to be regulated. So there is low levels of calcium ions in the cytoplasm compared to outside the cell, the mitochondria and the endoplasmic reticulum. Now more calcium ions can enter the organelles when the channels gets open. So that's how it can maintain the low level. So as earlier mentioned, that the cell membrane and the mitochondria have calcium ion channels or pumps. The third area or the organelle, which is little organs found inside the cell, where you can find these calcium channels, is endoplasmic reticulum. And endoplasmic reticulum, there's two types. There's the rough endoplasmic reticulum. This is just outside the nucleus, and as you can look at the image, you've got studied ribosomes where protein production takes place. Because the nucleus, what that does is where transcription takes place, so it converts the DNA to the RNA, and then the template is then being left the nucleus and it goes to the ribosomes where protein takes place. So the ribosomes is where proteins are made. And the rough endoplasmic reticulum is one of the areas where you can find ribosomes. So the main function of rough endoplasmic reticulum is to produce proteins, and also they're involved in sorting the proteins. The smooth endoplasmic reticulum, on the other hand, is where you can find the calcium ion channels or pumps. So it's involved in the accumulation of the calcium ions, also in the production of and the modification of fats. It's also involved in carbohydrates. So why is the concentration of calcium ions low in cells despite they have pumps? So just as a recap, that there are 10 to 20,000 fold difference in terms of how much calcium is being transported across the plasma membrane. So let's have a look at more closely with the first type of transporter, or the, that's more specifically, it's an antiporter. And what it does, it uses energy within sodium ion gradients to pump calcium ions across the membrane. So what that means is that the calcium ions isn't able to move across. It needs to utilize sodium ion, which is another type of ion, to be able to pump the calcium ions across the membrane using energy. So every time one calcium ion is out, there's 3 sodium ions coming in. So there's like a form of an exchange. Yeah? So some ions are coming in and other ions are coming out. Now you have these enzymes called ATPases, and what they do is they use energy by splitting or hydrolyzing ATP in order to pump calcium ions against the gradients. So earlier on we discussed what diffusion is. So diffusion doesn't require energy because it's down a concentration gradient, so it's from a high concentration to a low concentration. But when you're going against the concentration gradient, you need energy to do that. So a gentle reminder that ATP is an abbreviation of adenosine triphosphate, which is a type of energy source. The second type of transporter, which we introduced earlier, was the hydrogen calcium antiporters. So with this one, they actually also use energy as ATP, but they use their sodium hydrogen gradient pump to be able to remove the calcium ions across the membrane. So you have one sodium ion in and then one hydrogen ion out, in this case over here. So as you can see, the calcium ions are unable to move independently across the membrane. It's facilitated via these antiporters to help move the ions across the membrane. So the electrochemical gradients allow the cells to control the direction the ions move across the membrane. So let's look at the second example, which is CAMP. So CAMP stands for cyclic adenosine monophosphate. Now, it's a similar structure to adenosine monophosphate. So it has a ribose sugar, it has a nitrogenous base, and it also has a phosphate group. Okay, so it has one phosphate group. However, the difference between AMP and cyclic AMP is that the phosphate group isn't just attached to one position within the ribose sugar. If you look at the image over here, it's actually attached to both parts. So this image is taken from Alcitron. And to be able to produce cyclic AMP, you require an enzyme called adenyl cyclase. So adenyl cyclase utilizes ATP to produce CAMP. So once it's activated, what does CAMP do? So CAMP binds to an enzyme called protein kinase A, or CAMP-dependent kinase, so A kinase. And once that binding occurs, it activates it. And once you have your activated A kinase, this leads to phosphorylation of other protein targets because, as earlier mentioned, protein kinase A, they phosphorylate other proteins. So once these protein targets are being activated, the response varies and it can influence the CAMP levels. Now, the CAMP levels can be deactivated and converted into AMP via the enzymes Phosphodiesterase. So let's have a more visual image. So in this first image over here, you can see there's a ligand, it has an inactive receptor, and you have the plasma membrane. So the ligand needs to bind to the receptor in order for the receptor to be activated. So there's where you can see the activated receptor in the second image. Now once the receptor is being activated in the second image, you have an enzyme called adenyl cyclase. So the adenyl cyclase, you can see the structure of the ATP over there. So it has 3 phosphate groups, the ribose sugar and a nitrogenous base. The adenyl cyclase facilitates the formation of CAMP using ATP. Now, when CAMP is being produced, it needs to bind with protein kinase A to activate protein kinase A. Now, once protein kinase A is activated, that leads to phosphorylation of downstream proteins. Okay, so there's an addition of the phosphate group in each of these proteins become activated, and these protein targets are found within the cytoplasm. This then leads on to the transcription of specific genes for other target proteins required. So now cyclic AMP, when it switches on, you need adenyl cyclase. So ATP, it forms CAMP via adenyl cyclase. Now to turn cyclic AMP off, you would need phosphodiesterase. So the phosphodiesterase binds with the CAMP, deactivates it, and converts it back into AMP. Now let's look at the third example, which is deacylglycerol and inositol triphosphate. So inositol triphosphate is produced from membrane phospholipids. Now a gentle reminder that the plasma membrane or the cell membrane is made up of phospholipid bilayer. So bilayer means 2 layers. And a phospholipid has the blue circles over here is the hydrophilic phosphate head. And then you've got 2 fatty tails. Okay. So that's what you call a phospholipid bilayer. Now when we talk about a phospholipid, we're talking about one that just has the more or less the phosphate head and the 2 fatty tails. But let's begin more specifically with inositol phospholipid. Now, inositol phospholipid is a lipid fat molecule. So it has an inositol, which is a carbohydrate, as its hydrophilic head. And that is what acts as a secondary messenger because they are membrane bound and found near cell surface receptors. So the receptor in question we're looking at over here is GPRC, so G protein coupled receptors. Now there is a phospholipid called phosphodienoisotyl PI, and this is basically phosphorylated by kinase enzymes. And when it gets phosphorylated by kinase enzymes, it forms PIP and PIP2. So PIP stands for PI phosphate, so phosphodienoisotyl phosphate, and PI bisphosphate, so phosphodienoisotyl bisphosphate, so PIP2. Now, what happens is the alpha subunit of the G protein, so gentle reminder that the GPRC, it interacts with proteins called G protein. So when you have the ATP being joined onto the G protein, it activates it, and then it basically divides the G protein into an alpha subunit and a beta gamma dimer. Now the alpha subunit of the G protein, okay, this activates another enzyme called phospholipase C, so PLC. And the PLC is what then cleaves PIP2. So you can see one activates the other, and it's very sequential. Now, when the PLC cleaves PIP2, it forms 2 secondary messengers, DAG and IP3. Okay, so this is how IP3 is being produced from the membrane phospholipids and also DAG. But what happens next is very interesting because they differ in the mechanism in how they interact or transduce a signal. Because the DAG stays in the plasma membrane, it doesn't move anywhere. And what it does, it binds and activates an enzyme called protein kinase C. So it's still a protein kinase, but it's another subtype called protein kinase C. Now, protein kinase C phosphorylates cellular proteins. And as we discussed earlier, that when we discuss the term phosphorylate, it has to be, the phosphate has to bind to an amino acid within the protein to be able to phosphorylate and bind. And serine and threonine are the amino acid residues that protein kinase C phosphorylates within the target cellular proteins. Now the IP3, on the other hand, it doesn't stay in the plasma membrane. It diffuses into the cytoplasm. And when it diffuses into the cytoplasm, it directly goes to the calcium channel or pumps in the endoplasmic reticulum membrane. Okay. So when the endoplasmic reticulum membrane the calcium ion channels over there, when they open, it releases the calcium ions. Okay, so it releases the calcium ions. This is what continues what we call a cascade. So activation of the downstream proteins to cause a cellular response. So as you can see over here, is IP3 is a secondary messenger, but it also interacts with other secondary messengers such as calcium ions in order to transduce a signal. Now, Modulin, CAMP, is a protein whose role is to bind to calcium. So calcium ions doesn't only just need the antiporters to basically able to enter and leave accordingly, but also it needs to be bound with a calcium binding protein to be able to be regulated to ensure in terms of the low concentration within the cells, and also regulate calcium dependent pathways. For example, when you want to break down carbohydrates within the liver cells, that is required. So what do I mean by that? So say for example you've got high levels of sugar, glucose, inside the blood, and what happens is that there's a hormone called insulin, and that is a type of protein hormone, and it sends a signal to the muscle cells and the liver cells and says, hey, there's a high levels of sugar within the blood. We need to convert it. So they're going to convert the sugar level to more of a storage type of sugar compound called glycogen. And this is what helps to lower the levels of sugar within the blood. Now, when there is low levels of sugar in the blood, the opposite occurs. So you have another type of protein hormone called glucagon. And the glucagon says, hey, there's low levels of sugar within the blood. So then the glycogen, which is the carbohydrate, needs to be broken down within the liver cells to help return it the normal blood glucose levels back to normal. So on the outside you may see this is a hormone, but you can see that there is lots of interaction between the ions and the molecules working together to be able to facilitate such a response. So that leads on to cellular response. So cellular response depends on the type of receptor, the signal transduction, and the target cellular proteins. So, for example, calcium ions is such an important secondary messenger, and it requires proteins to perform the following functions: muscle contraction, cell growth, hormone release such as insulin, the transmission of nerve messengers, also the secretion of enzymes or production of enzymes are involved in the digestive system to break down food from large to small. Also, it's involved in plants. So plants, particularly within the leaves, they've got small holes or small pores, and these small holes or pores are known as stomata. So this is how oxygen and carbon dioxide are able to diffuse in and out, okay, and also how water can also leave. There's also cell elongation, and that occurs in response to light. So how the plants respond to light or respond to gravity, that is also involved by calcium ions. Now, calcium ions are also involved in cancer cells. So, gentle reminder that the hallmarks of cancer, You've got evade apoptosis, unlimited replication, sustained proliferative signals, evade growth suppressors, avoid immune destruction, reprogramming of the energy metabolism, angiogenesis, invasion metastasis. And then you have contributing factors such as genomic instability and tumor promoting inflammation. And collectively, this is what can cause cancer and metastasize. Now, in relation to calcium ions, the secondary messengers, calcium ions actually regulates many hallmarks of the cancer. More specifically, self-sufficient growth signals, evading apoptosis, its sensitivity to antigrowth signals, angiogenesis, invasion and metastasis. So these hallmarks have been developed by Douglas and Hanahan and Robert Weinberg. So you can see how the hallmarks of cancer and the calcium ions interrelate. So let's progress on to transcription factors. Now, transcription factors, these are proteins that are more or less involved in the transcription of DNA to RNA. Now, in order to produce proteins, it's a two-step process, transcription, translation. So DNA needs to be unwinded to form the RNA, and that step is known as transcription. Now, the RNA template then leaves the nucleus, travels to the ribosomes to translate that information there in order to produce a protein. So the transcription factors focuses on the transcription, and it regulates the genes expression of these target proteins, and this essentially affects the cellular response, such as differentiation to specialized cells, development, and immune system. And there are approximately 3,000 transcription factors that regulate about 23,000 genes or so encoded by the human genome collectively, and that is fascinating. So some transcription factors can bind to specific areas called promoter sequences, and these promoter sequences are near where transcription starts. So transcription has a starting point. So this is where some transcriptors can bind onto. Now, other transcription factors, they bind onto an area called regulatory sequences. Now, these areas consist of many base pairs that are near the target genes. Now, base pairs, you should be familiar with this, whereby we discussed this in one of the previous lectures. There's 4 types of bases: adenine, thymine, guanine, and cytosine. And if it's RNA, it's uracil instead of thymine. So you have complementary bases, so it's ATGC. So when we refer to base pairs, this is what we're referring to. And if it's RNA, then it's AU instead of AT. The activity of a transcription factor can be regulated by production of particular cell types only, and also through phosphorylation. So let's look at the types of domains in transcription factors. So here you can see there's a structure of the transcription factor there, and you can see there's lots of domains. So you've got the dimerization domain, the DNA binding domain, DBD, you've got the ligand binding domain, the transactivation domain. and then you have the C-terminus. So the N-terminus, that's referring to the amino group, or the amine group, and then the C-terminus, that's referring to the carboxylic acid group. Because if we look at the structure of a protein, it's made up of amino acids, and then amino acids, they have a amine group and they have a carboxylic acid group, and then they have an R side chain. So this is why it's called N-terminus and C-terminus. Now the DNA binding domain, DBD, it has 4 subtypes: so helix, turn, helix motif, leucine, zipper motif, helix loop, helix motif, and zinc finger motif. And a zinc finger motif is approximately 30 amino acids long that is surrounded by a zinc atom there. And a zinc is a type of metal. And collectively, these 4 subtypes create a form of a conformation change which helps the DBD with the binding of the DNA. Okay, so the DBD helps the transcription factors to specifically bind to the DNA sequences by recognizing these areas, and especially in the major groove of the DNA. So we said the DNA structure is a twisted structure, and when you have a twisted structure, you have like a larger gap and a smaller gap. The larger gap is known as major groove, the smaller gap is known as the minor groove. So the DBD directs the transcription factors to bind onto the major groove, to the specific areas that contain the sequences required. And because of how collectively conserved and how they work well together, it's more or less helps to classify transcription factors into different groups. Now the effector domains, there's 3 types. Now there's some transcription factors that have transactivation domain, there's some that have repressor domains, and some that have both, i.e. bifunctional domain where they can activate or repress gene transcription. And the function of these effector domains is to help modulate transcription activity. And how it does that is to bind with cofactors, and this what causes changes to the histone proteins. Within histoproteins, you can only find them in eukaryotes, i.e. plants and animal cells, and this is what keeps the DNA compact. And there's also DNA methylation, so this is when you have DNA being attached to methyl group, and methyl group consists of CH3, and also it activates RNA polymerase II, which is an enzyme that is involved in transcription. Another type of domain is dimerization domain. So dimerization domain, this helps transcription factors that work in pairs or in dimers to facilitate transcription. So there are some transcription that work in pairs, either as homodimers or heterodimers. Homodimers means the same, heterodimers means different. So they can bind to DNA as 2 transcription factors that are the same working together, or 2 transcription factors that are different but they bind to the DNA. There is also ligand binding domain. So some transcription factors only function upon binding of a ligand, and therefore they require the LBD. So by the end of this lecture, you should understand that the first messenger in the cell signaling pathway is the ligand. The secondary messenger helps transduce the signal to elicit a response. So calcium ion channels are found within the mitochondria, the endoplasmic reticulum, and the plasma membrane. It is regulated to maintain its low concentrations in the cell. It binds specifically to the calcium binding protein called Modulin. Now the CAMP plays a key role in signal transduction pathway. So it's activated by an enzyme called adenylyl cyclase, where it then binds to protein kinase A, and this is what activates more proteins in the signal cascade. And the CAMP is activated by Phosphodiesterase. Now DAG and IP3, they are also secondary messengers that are involved in signal transduction. How? Well, for in the case of DAG, it's in phosphorylation of downstream targets via protein kinase, and IP3 it stimulates other secondary messengers such as calcium ions. Transcription factors, they regulate the expression of genes which affect the cellular response, and they have particular domains that help facilitate this. Here are a reference list for further reading. Please go ahead and if you are interested, have a read and further. Next week we'll be moving on to lecture 6. So now we've introduced the 3 steps of cell communication: receptor activation, signal transduction, and cellular response. Now we're going to be more specific with different examples of signaling pathways. So in next lesson, hopefully we will look at what the key steps are involved in the GPCR signaling pathway and what happens when it's dysregulated in cancer cells, examples of cancer that is involved, the causes, and how the normal pathway is affected within cancer cells. I hope you have benefited from this lecture, and if you have any questions, please do not hesitate to email me. Take care and enjoy the rest of your week. Bye for now.